Thank you so much for the invitation. It's really a pleasure being here and discussing all these very interdisciplinary concepts, and it's very refreshing for my mind. So it's my first time in Japan, and I'm really happy to see that you also have your own Eiffel Tower. Maybe it's not as nice as ours, but in general, what I can say is that you have a much better weather than we have in Paris at this moment. So who am I? I'm an evolutionary microbiologist. I work at the Institut Pasteur, and I guess what we are interested in, I'm not at all a specialist on the origin of life, but we're more interested in whatever happens after the LUCA stage. So basically, we can say that we are interested in resolving the phylogeny and evolutionary relationship among the three domains of life after the LUCA, so the evolution of life, and then using this robust reference phylogenies of species to map and understand the emergence and evolution of specific characteristics of life. So for someone that does this, these are really exciting times, because I've never seen a field that is changing so much and so rapidly over the last few years. Starting from this little tree, as Sean told you, 40 years ago by Carl Woese, who did it by using this molecule, which is the RNA that is embedded in the ribosome, in the small subunit of the ribosome. The progresses in obtaining genome and sequence data from a large diversity of microorganisms, combined with the new technologies to analyze them, they're really revolutionizing completely our view of the tree of life and how we think about the diversity of microbes, and changing a lot of paradigms that we thought they were absolutely settled. And in all this revolution, the Archaea have been instrumental. And what I will talk about today, it's really a dramatic example of an expansion of a clade and what it makes on the tree of life. But what I think is important is that the tree of life is changing, and this is normal. It was never meant to be a static structure. And I hear a lot about people saying the tree of life is wrong, the tree of life is wrong, now we have different trees. But I think it's very important to call it always the was tree. Even if we have 1 domain, 3 domains, 50 domains, this is the was tree. It's the tree of life that was made by the contribution important of this person. So I guess what I will be talking about, even if you don't get all the things that are too specific, is in general 2 types of emergence. One type of emergence is how all these new sequences and all these new methods brought up to the surface a diversity of microorganisms that we did not know it existed. So that's that kind of concealing, becoming visible, something that has been concealed up to here. And that it's instrumental to change completely our perspective on how we image the origin of specific characteristics and their evolution. So the example I will make, it's really about the Archaea. So this is my 3 seconds Wikipedia slide on the Archaea. I think it's important to explain what they are. Maybe you know, but this is still the domain that is less known, less studies. This is normal, first of all, because they are the ones that have been discovered more recently, only 40 years ago with respect to bacteria. And the second is because we don't have pathogens in the Archaea, so there are much less interest in studying them. But nevertheless, this is the way they are perceived in the public opinion, in the public view. So they are called extremophiles. They can thrive in very extreme environments, such as reaching very high temperature, pH close to 0, and huge amounts of salts, for example. So if you look at them, the Archaea basically look pretty much like bacteria. But what is, they're round, little, just like bacteria. But what is really important is that if you go at the molecular level, the Archaea are really much closer to us than they are to bacteria. This is really stunning. And in particular, these are not little characters. There are the structures and the processes that are really important to the cell, especially the ones that are used to transfer the genetic information from one cell to the other. The most dramatic example is DNA replication, which is absolutely very, very conserved between archaea and eukaryotes, and it's completely different from bacteria. Then we have transcription, where you can see that archaea and eukarya have these machineries that are much more similar, much more protein-rich with all these components. And even in the ribosomes, there are proteins that are uniquely shared between archaea and bacteria, archaea and eukaryotes, and not bacteria. So not only are these important differences between archaea and bacteria, but also archaea have their own peculiar characteristics. One in particular is their cell envelope. Bacteria, for example, have a very, very uniform cell envelope with a layer of peptidoglycan. Archaea are much more diverse in their cell envelope. But one thing that is really, really stunning is in the lipid composition of the membrane. So if bacteria and eukaryotes, the lipids of the membrane are esters that are linked to a backbone of glycerol through ester linked. In archaea, the link are ether, and the glycerol phosphate, the backbone is 1-phosphate. So here we have lipids that are, the glycerol backbone is completely, the stereochemistry is completely opposite. We talked about chirality yesterday. And the enzymes that make these lipids are completely different and than families. So this is what also is called the lipid divide in the tree of life, and we don't know much about the evolution of this. So this was the situation of the diversity and phylogeny of the archaea in 2005, based on 60ness. So you can see there are two major phyla, the Crenarchaeota, called like this because of most are thermoacidophilic, so for origin and spring. And Eukaryote are much more diverse. So they have different metabolism and live in different environments. But so you can see two things from this tree. The first one is that we had very few archaea that were in culture in the lab. These are the only ones that are the black boxes. All the rest at the time were archaea uncultured. It means that we have not them in the lab, and they are represented only by their sequences, the 60ness sequences. And the second thing I want you to notice is that this tree is what we call the tree that is completely unresolved. So you cannot really tell the evolutionary relationship among the different lineages. And this is something that happens in deep phylogeny and is due to one particular fact is that the 60ness only cannot resolve the very deepest divergence in the tree of life. There are two reasons for this. One is radiation, so there is not enough phylogenetic signal, meaning that at the onset of the emergence of a clade, you have all these lineages separating very rapidly, and so there is no time in the sequences to record the changes that can give us information on this event. The second one is what we call mutational saturation, meaning that during evolution there are changes in the sequences, and sometimes you have multiple changes that erase the changes before, and so even if you go a long time long time back in time, this is become really important, and in the present day sequences you have only a tiny fraction of all the changes that have occurred. So one thing to overcome these problems in reconstructing deep evolution is what we call the phylogenomics. This is a very fancy name to say something that is very simple. We reconstruct phylogenies not by using one single gene, but by using contexts of the genomes. There are many different methods, but the ones that work really well to reconstruct the phylogeny of species is the one that are based on orthologs, means genes that have been inherited vertically, we say, through speciation, and can be used to retrace these speciation events. So we have this method that are called super trees, super matrix. I mean, it's really funny, but they're really super in what we do. Basically, we use many genes combined to reconstruct evolutionary relationship. The one that really works really well is what we call supermatrix approaches. So meaning that you put all your gene alignments one by one to make a mega gene. And in this case, you amplify enormously the phylogenetic signal, and you can reconstruct better the evolutionary relationships. But then, of course, to do this kind of methods, we need more genomes. And I told you that the Archaea for a long time, where a lot of Archaea were not cultured, and that's very difficult to obtain sequences in that case. And this comes very handy. Is new methodologies to obtain genomes from environmental samples where you have complex community of microbes. It's called genome-resolved metagenomics. So in a very simple way, you have a sample, environmental sample, where you have lots of mixed community of microorganisms. You sequence everything. You obtain the sequences of everything. And then you use particular methods to assign each genome, each sequence piece to a different genome, because each individual has a specific signature of nucleotides, of tetramer of nucleotides, and so we know how to assign one specific sequence from one genome to the other, and we have those very fancy graphical maps of this, where each color spot represents a genome of an organism. So this is very interesting, and this is what happened with the Archaea. We went really from the first genome that was sequenced in '96, when more or less the first bacterium genome was sequenced, but then the Archaea lagged behind really a lot. And you can see in the really last 5 years, we had such a dramatic increase in the obtention of sequences from the Archaea that it's really stunning. And most of them come from uncultured lineage. So we went from that little tree to something like this, which really gives you an idea on how incredibly expanding is the tree of Archaea. And I like to cite this sentence by Gary Olsen in '94, when we started doing environmental microbiology, saying that overlooking the Archaea was equivalent to surveying 1 square kilometer of the African savanna and missing over 300 elephants. But now I can say that it's even more important now. So now we have a fantastic base to study more the diversity and evolution of the Archaea and the origin of all these peculiar characteristics. So the first, I will just make 3 examples on this. The first one is that now we are spotting new characters in the Archaea that we didn't know they exist. So these are the Tom Archaeota. It's we named this phylum because it was not grafting within the Crenarchaeota and the Eukaryote. And this is the first evidence of nitrification in the Archaea. They're aerobes, a major part in the nitrogen cycle, and they're also present on our own skin. So not only the Archaea are not only extremophiles, but can also be major players in the global biogeochemical cycles of the Earth. The second thing I wanted to tell you about is that this is something really funny that emerged in the Archaea, a new way of life that we didn't know it existed. So this is Nanoarchaeum equidans, and is a little archaeon that has chosen the most curious way of living. Basically, they live in hyperthermophilic environments attached to another archaeon, Inicoccus hospitalis. So the little cells are Nanoarchaeum, and Inicoccus is the host. So it's been identified completely by chance, and they're very small cells, tiny cells, and with the small cells causes also a very small genome that has lost a lot of very important genes. So they are really dependent on the host. Otherwise, if you detach from the host, they cannot survive. So this is what we call the epibiont, in a way, living on the surface. And when we try to understand the origin of these Nanoarchaea, we are encountered a little problem, because they are very divergent. So they form a long branch in trees, and we don't know where they have a very unstable position. Most of the times they go at the base of the archaeal tree, and sometimes no. And this is something important in phylogeny when you try to place very, very long branches. This is an artifact that is very well known. When you have a situation in a tree where you have one branch that is substantially longer than the others because it's have a lot of mutations, most phylogenetic methods will put it at the base of the tree because it will be, by definition, attracted by the long branch of the outgroup, which is by definition the most distance. And so in this kind of artifact, a character might appear older than it actually is. But what's important is that now we think there are many, many more of these nanozized archaea, and we know that only by sequences, environmental sequences. So they seem to have all the same characteristics, and this huge expanding clade, it's something really important in the archaea. So at this moment, maybe this is an emergence of a new lifestyle by subtraction instead of accretion of new characters, which is interesting. And I don't know whether they all together or they emerged many different times independently. And it's even more interesting because it opens up a lot of questions on how we got to this extreme genome reduction. The third example I wanted to make is about methanogenesis. So methanogenesis is a complex metabolism that only archaea can do. Bacteria cannot do this. It's the production of methane through the successive reduction of carbon dioxide or small carbon molecules like methyl, acetate, for example, methanol. And it's very, very important metabolism in the global carbon cycle. Methanogens have been studied most of the time from the rumen cow, but it's much less known that they are actually also present in our own body, where they live happily within us. I hope not too happy at this moment. And what's important of methane is that it's one of the most ancient metabolisms. It might have been responsible for the reason why the Earth was not glaciated like a long time ago. Ueno, who I know works at the ELSI, found the oldest trace of biological methane at 3.5 million years ago. And also, maybe methanogens are important for the search of life in the solar system, because recently some colleagues in Vienna managed to grow Methanococcus okinensis, with, I think it's Japanese, in conditions that mimic the one found in Enceladus. So I'm not sure whether these are actual archaea on Enceladus, but it's important. So for a long time, we had this image of the archaea. And so you see there are only two little classes, class 1 and class 2. And we thought that there was really one origin, not very early in the archaea, with a few losses of this metabolism. But things started changing a little bit. In 2013, we found a new group of methanogens that was not in class 1 and class 2, and the peculiarity of being strictly dependent on methyl compounds for methanogenesis. So, okay, this is interesting, but this was just anticipating a deluge of new methanogenic lineages that appeared in the last few years. And some of them are even not methanogens anymore. They use other compounds, like, for example, butane, and these are, it's very exciting. And very recently, by digging into the databases where people store metagenomic data, we found an incredible diversity of new lineages. They are really, really everywhere. They are in all the archaeal trees. So we think that this might indicate the methanogenesis is really ancient in the archaea, and it's one of the foundation metabolism of this domain of life, and that was lost repeatedly or changed and mutated repeatedly during the evolution of the archaea. And then another interesting thing that we will look at in the next few years is an emergence twice of adaptations of archaea to the human body, which is something that it's not very well known. So one important thing with all these new genomes of archaea is that we discovered also new numbers of characters that are normally known to be present exclusively in eukaryotes. And this is really exciting. I will not get into the detail, but just to tell you that most of these characters have nothing to do with the characters that we knew before, but have something to do with the cell division or cytoskeleton of characters that in eukaryotes are important for the division of the cell, the structure of the cell. So this has relaunched completely the idea of the relationship between archaea and eukaryotes. So you need to know that the eukaryotic tree has also dramatically changed over the last few years based on this phylogenomic analysis and multi-genome analysis. So we occupy only a tiny part of the huge diversity of the tree of eukaryotes, which is dominated by microorganisms. But this tree also was instrumental to try to understand what the last eukaryotic ancestor looked like, which is called the LICA, last eukaryotic common ancestor. Basically, you find, you map characters on the tree of life, and if you find them more or less distributed, you put them into the LICA. But when you do this, you get to an image that is really, really frustrating, because this ancestor had basically everything. It was a fully fledged eukaryotes. So he had the nucleus, splicing, sex, mitochondria, whatever, even important metabolism. So at this moment, we are really stuck at the point where we have everything at one point, and we need to understand how these characters were assembled over time, which one comes before the other. So we need to go deeper into the tree of life, and when we go deeper into the tree of life, this is where we bump. We bump on the archaea. This is the tree of life rooted with the archaea, and this is another important notion that in this tree of life, we have eukaryotes and archaea. There are sister lineages, and this means basically that the ancestor was not an archaeon, not a eukaryote, it was something different, and eukaryogenesis, what we call the making of the eukaryotic cell, occurred on that very long branch. So this, of course, was a very important result, but it was based only on one gene at the time with very simple evolutionary models and not much data. And now the things are really changing because there are three important things that we have. The first one is that we have models that model the evolution of sequences much, much better. This one in particular is made by colleagues in France. It's called CAT, and it's a Bayesian model that allows that your amino acids in the alignment can vary each one according to their own substitution matrix. So each one is modeled as a process. And this is really much more realistic, and it's very useful to resolve very deep evolutionary transitions. Then, of course, it's very parameter rich, it's very computer intensive, and now we have the computers to search the tree space and use this kind of powerful models. But another important thing is now we have much more sampling from the archaea, which maybe can give us some information. And this is exactly what happened when we apply this kind of methods. In 2008, by reconstructing a universal tree based on with all these genes, you had eukaryotes basically moving from outside of the archaea inside the archaea. So they're branching really within. So you have the LACA, the last archaeal common ancestor, and a split between Archaeota and all this other group that is called the TAC for Tom Archaeota, Egg Archaeota, Cren Archaeota, and Cor Archaeota. And there is an unresolved relationship between eukaryota and this group. So as you see, all these analyses were done with universal genes. Of course, you need to reconstruct the universal tree of life for that. But recently, we did a little different method using non-universal trees, but analyzing the domains 2 by 2. And we flipped a little bit even more the deepest branches of these three. So basically now we have, yes, of course, eukaryotic stemming from within the Archaea and next sister to this group of the TAC. But we also flipped sensibly the first branches of the very archaeal tree, and this also has consequences on how we estimate the LACA, and in turn we go down to the LUCA. So you see the tree is really changing completely shape little by little and changing perception on all these ancestor points. So basically, if we go and explore more around this TAC group, maybe we will find some kind of missing links or something that will give us some more information on the emergence of eukaryotes. And this is exactly what happened. The group of Thais Ettema in Sweden went to sample more genomes from the TAC group to see whether it was giving some information. And this is exactly what happened. It went into a deep marine sediment, 3,000 meters below the sea level, and at a site that is called Loki's Castle. So he got a genome of an archaeon that is called Loki archaeon. So it's pretty much an archaeal genome, but it has some particular genes that we didn't think they were present in the Archaea before, and they are very, very eukaryotic-like proteins and cytoskeleton elements. And when you reconstruct the tree, you have eukaryotes really branching very closely to these Loki Archaea. And recently, they obtained even more of this group, and they are giving all these fancy names of gods, of northern gods, Thor Archaeota, Odin Archaeota, Heimdall Archaeota. And what is interesting is that this group really looks like it's enriched in all these characteristics that normally we thought they were present only in eukaryotes, and in particular, cytoskeleton methods. So what's important to understand is that now there are really a few groups that are trying to get their hands on these Asgard Archaea, because these are only sequences. We have never isolated them in the lab. We don't know what they look like. And to me, it's very interesting to understand what these characters are doing in here. So now we have this situation, and we have eukaryotes that emerge from an archaeon. So we have a starting point. And this is really exciting to try to understand how all these characteristics emerged. But we also have this kind that you need to understand that there is still a very, very long branch going from the Archaea to eukaryotes, and it involves also a lot of extinct branches in this Archaea. And we are in this kind of twilight zone of emergence. It's not a dramatic one, but it's something more progressive, which we don't know really what happened. And what's important is to understand in particular, the emergence of two characteristics. One is the cytoskeleton that would have allowed this organism to pick up the mitochondrion, which is an important founding event during the evolution of the first eukaryotic cell, or whether mitochondrial endosymbiosis was really the first founding event. But we get also into a kind of philosophical question. When do you stop being an archaeon and you start being a eukaryote during this process? To me, it's probably when you start developing a nuclear envelope, because the nuclear envelope is what defines the eukaryotes. But maybe not. But personally, what I think that we are finding all these proto-components of cytoskeletal elements in these archaea, I think that the cytoskeleton started developing first, and then you had the possibility of having larger cells, phagocytosis, and then mitochondrion, and then off you went to all this innovation leading to eukaryotes. So because we are eukaryotes, of course, we are really, really interested into this eukaryogenesis. But what I would like to point out in the last two slides is that we should really also think about the very emergence of archaea. Why they're all obsessed about eukaryogenesis, and we think that prokaryotes are much more simple and not interesting. But the emergence of all these characteristics in the archaea are very, very puzzling to me. Why are they so different from bacteria? Now what they know with the LACA is that was very likely anaerobic, methanogenic, possibly thermophilic. But then why all these characteristics of the cell are so dramatically different from bacteria? And so what we really need to go down another level in the tree of life and start to understand the evolution, the divergence between archaea and bacteria. And this is the tree that Sean was showing you. The bacteria is the most incredible challenge for deep phylogeny in the next few years. We have all this new diversity, this enormous number of genomes and sequences. The vast majority is uncultured, and as Sean was saying, this is this huge group emerging recently in the bacterial tree, which is called the candidate phyla radiation. And what's interesting in this particular group is that it looks like they are very similar to the nanozized archaea that I was showing you about. So they look like they are epibionts, very, very small cells attached to other bacteria with very, very reduced genomes. And there are a lot of them. They are everywhere. We just didn't know they existed. So to me, now, I'm really interested in understanding, is this the first form of life, or these are not at the right place, and they emerged independently in archaea and bacteria. So with this, I would like to thank you for your attention and remind you that all these exciting novelties on the archaea field will be discussed in the Gordon Research Conference that I'm organizing with my colleague Finn Werner in the beautiful place that is called Les Diablerets in Geneva. And in particular, this Gordon conference will be focused on the place of archaea in the tree of life and the comparison with bacteria and eukaryotes. So please do come if you're interested in knowing more about this. Special thanks to my group in Pasteur, which is a nice mixture of people with experimental and bioinformatic capacities. And also a lot of thanks to Madame Sato for a lot of assistance before and during my conference, in particular when I left my computer nicely at the airport in Paris, and I realized it when I arrived here. Sean, of course, Tomo and Koske for being such wonderful hosts and for all the food. Thank you for your attention.
Okay, thanks, Simonetta, for the wonderful talk. We're a little behind schedule, I think, but let's have a few questions for Simonetta. I have the orange box ready.
There's one over there.
Jake Hansen, Arizona State. Last year I heard of a few papers talking about a universal core metabolism, and then the ability to extrapolate that to a location on Earth where life may have originated. Can you say anything about that? Is that something that is generally accepted?
Oh, yes. So there is a big challenge in extrapolating metabolisms in the last universal common ancestor. In general, what you do, you try to find metabolisms that are shared between bacteria and archaea, because now the eukaryotes are not interesting anymore, and you put them in the ancestor. There are a lot of challenges because there are not many genes that have remained in this ancestor. Most of them have been transferred, most of them have been lost, and even the methodologies are really difficult to study. I think you're referring to the work of Bill Martin and other people. So they inferred more or less 300 genes in the ancestor in Luca, and a metabolism that is based on hydrogen and completely anaerobic. So at the moment, yes, this is more or less. It might be inferred in that way, but there are a lot of caveats with those kind of methods. What I can say for the moment is safely the most ancient metabolism in the ancestor of the archaea, but in the Luca, I'm still not really sure about what. Okay, it's partial vision.
Take one from the top here, from Batul, about carbon fixation mechanisms in the archaea. Do you believe any particular one of them preceded any of the others?
The Wood-Lundell pathway.
There we go. And Caleb, did you have a question?
almost killed someone there. It was a beautiful talk. So this is going to be a very ignorant, stupid question from someone completely outside the field. Horizontal gene transfer, how much does that mess up these phylogenetic trees? Do you have a sense of that?
Yeah. It doesn't. In the way that you need to understand that these trees are very representation, kind of simplified representation that we have, but you need to imagine them with lots of transfers all the time. So these are kind of reference trees on which we can map horizontal gene transfers. So there is horizontal gene transfer and phylogenetic trees of species are not in opposition. They're one on top of each other. It's just that we represent them like this. And there are transfers, but not as many to completely erase this phylogenetic signal.
Thanks.
Back to the board. David Baum has a question. Where are the CPR bacteria?
Why should they be progenitors of mitochondria? I don't. Right. Okay. So for the moment, they are right at the base of the bacterial tree. So what I'm interested in is try to understand whether if I do the tree differently with different methods and different markers, maybe they move up somewhere else. But I'm not sure whether they will go up to the alpha proteobacteria, but mostly in the deepest branches. Most of them are anaerobic.
Okay, then I'd like to thank Simonetta again. I'm sorry we have to keep moving, but we can keep discussing later on. I'll note that we're reanalyzing the data from that 2016 Vice paper right now, so if anybody wants to talk about what might have been in the Luca, let's talk about that later. That'll be fun. And thank you again, Simonetta. Thanks for coming too.